Hello, everybody. Mm.
Hello.
Okay, so I'd like to thank Brian Pinello for telling me about the Ethernet, which I probably knew about before you were born and hadn't thought of, but I'm directly in now, so I should not have any trouble. Um, Also, Monday or Tuesday, because I cut off so many times, all it really recorded was the first 3 minutes until the first time it cut off. So there really wasn't any recording from Tuesday. So hopefully we're not going to have that problem today. How many people we got? 39. Wow, I feel popular. Or is it just because it's a required course? I don't know, one or the other. Let's go ahead and get started. What the hell? That's what we're here for, right? Learn a little cell biology. Come on, screen share. Here is my PowerPoint. Okay. I had this done before I opened the Zoom, I know, but I am lazy. People are still coming, so it's not the end of the world, right? Okay, so I put my notes up probably about 10 minutes ago, so they're there if you want to follow along with it. So today we're going to be talking about microfilaments. Microfilaments. Oh, you've been waiting years to have the opportunity to get into cell biology and talk about microfilaments. So here we are. Today's the day. So here again is that immunocytochemistry thing where you can see you're lighting up all of the proteins you're looking at. I believe, not 100% sure, but I think the white stuff is all actin. So those are microfilaments in the cortex around the cell. And then this extension here is the lamellipodium. So this cell must be stimulated to start to crawl. So it's got branched microtubules or microfilaments in a lamellipodium out there. So we'll talk about that today, if you don't mind. Okay, so microfilaments. What the hell are microfilaments? Microfilaments are small filaments, right? Small, like long strings of protein polymerized together. Now, the protein involved is actin. You probably know actin from skeletal muscle and skeletal muscle contraction. Well, actin is all over the place. There's a lot of actin. That's one specific kind of actin found in the thin filaments of skeletal muscle. And we'll talk about that in the next chapter. But here is, in general, a microfilament, and it's made up of actin. Actin can have either GTP or GDP, or no, I'm sorry, ATP or ADP bound to it. In order to polymerize, very similar to microtubules, in order to polymerize, it has to have ATP on it. So once you've got ATP actin, it can be polymerized onto an existing microfilament, and it will get longer and longer and longer. It will then hydrolyze that ATP into ADP, which makes it less stable. So similar to microtubules, and the whole idea is you have to have charged monomers to add them. Once they're added, they'll turn off so that the structure can be taken apart again. So this structure also has a directionality to it, like we said in like microtubules. You've got a minus end and a plus end. Again, they're not charges, they're just one end versus the other. The minus end is also called the pointy end, and the plus end is the barbed end. And the reason they call it that, they say that in the book a little bit. I've never heard of it outside of your book, but you should probably know the nomenclature just in case it's used somewhere else. So it's based on the fact that if you took the molecule myosin and cut it, so all you have is the head of the myosin and a little bit of the tail, you have this kind of L-shaped thing. You put that in with a whole bunch of microfilaments, and it will bind to the microfilament with the tail shooting in one direction, and it will always shoot towards the plus end. So this end ends up pointy, this end ends up barbed. So that's all. I mean, I will never ask you that on a test. I'm just letting you know where the nomenclature comes from.
Professor,
So,
may I ask a question?
yes.
When you say pointy, do you mean like sharp at the end? And then when you say barbed, do you mean like still just also like what do you mean by
Well,
barbed? Is there a difference?
well, yes. Think about it like an arrow. Like there's the pointy end of the arrow, and the other end has the feathers, and they're like the barbed end. Okay, so the myosin when it attaches is like the feathers pointing backwards.
Oh, okay. That makes
Okay.
sense. Thank you.
Okay.
Thank you.
All right. So monomers are called G-actin, and the polymerized actin is called F-actin. Now, the reason for that is just historically, actin was discovered and characterized back in the 50s, and they didn't really know that it was the same protein polymerized versus depolymerized. They just said sometimes it's kind of in a globular form where it's individual molecules, sometimes it's fibrous. So that's all. And again, that's another kind of nomenclature thing that I'm never going to ask you. It's good to know it though, because you see it in different places. I'll always refer to it as polymerized versus monomers. When it does polymerize, it ends up with this coiled coil structure. It's kind of like you took a strand of pearls and twisted it. So you've got two strands wrapping around each other. And that whole idea of a coiled coil structure, we'll see a lot in protein structures, especially structural things, because if you had just one string there, it would be a lot more flexible. It would be easier to break apart. The coiled coil gives you a little more structure to it. So we see that theme played over and over again in a lot of different proteins. Okay, so those are your microfilaments made up of monomers of actin, polymerized to give you a long fiber that actually looks like this. This is your space filling model of the actual proteins. In terms of the monomers, the actin, there are a shitload of actins in the world. In your genome, I mean. There are, what do I have? Over 20 genes, or it's either 20 genes or 20 families of genes. So you've got a lot of different actin genes in your genome, which shows you how important it is, probably. I mean, that means, if you lost one, you would probably be okay. And these superfamilies are broken down into two different types of actin. You've got alpha actins, which are in skeletal or in muscle, right? Skeletal muscle, cardiac mostly. I don't think they're in smooth muscle. I think it's just skeletal and cardiac. And then you've got beta and gamma actins, which are non-muscle. So every cell has them, including muscle cells. So those are the actins that you'll see in the cortex of the cell that are, like I say, every cell has got a cortex. So every cell will have that. So that's it, muscle actins and non-muscle actins. I guess that's it for slide 24. So that's the general structure. And then we have to talk about how it's put together in cells. Hang on one second, I'll close my door. Got someone tiling my bathroom today, and he's learning about cytoskeleton while he tiles my bathroom. Okay, so we'll talk about parallel arrays, antiparallels, and gel mesh. So one type of arrangement are these parallel arrays. Okay, so you've got you've got them all going in the same direction, and they're going to be cross-linked by proteins. Like here, you see these structures in really, as far as I know, only two places, in microvilli and Filopodia. So microvilli are found on the villi in the small intestine. So they're just little projections of the plasma membrane that increase the surface area of your small intestine. So the membrane of that microvillus would be packed with transporter proteins to bring sugar in, bring amino acids in, again, making your smooth intestines more efficient. And then filopodia are involved in cell crawling. We'll talk about that in the next chapter, but when a cell starts to crawl, it puts these little filopodia out, like little fingers reaching out to grab onto things. And there's a little bit of stiffness to these because, you know, you've got your microfilament, which is kind of a loose structure, but then when you start cross-linking them, they get a lot more stiff. So especially in your microvilli, anything going down your intestine would just rip them off if there wasn't a little bit of strength there. So the whole idea of this microvilli is you've got a little bit of stiffness there with those microfilaments cross-linked, and that's going to hold the plasma membrane in place. Plasma membrane, keep this in mind, I say this a lot, but phospholipids will not stay in a plasma membrane structure unless they're held that way. If you took all the cytoskeleton out, your plasma membrane would collapse into vesicles. So this structure is holding that membrane in place. The cross-linking proteins here in microvilli and filopodia are fibrin and vilin. I think alpha-actinin also. Let me look at this real quick. Yeah, and alpha-actinin. So those are some of the other proteins that are involved in cross-linking. So here, filamin we'll talk about later. So these are just the straight cross-linking things. Fibrin, vilin, alpha-actinin. Okay. And here they are again. See, alpha-actinin is keeping them a little bit further apart. Fibrin is another cross-linking protein, and it keeps them a lot closer together. So this would be a stiffer structure than this. And then filamin we'll talk about in a second. Okay, so those are your parallel arrays, and mainly just used for structure to hold a plasma membrane out there like that. The next type of structure are anti-parallel arrays, and we see these in stress fibers. Stress fibers are really important for the movement of the cell. Again, chapter 14, we'll talk about it soon. But basically, they stretch the whole length of the cell and overlap each other in an anti-parallel kind of arrangement. And when they slide past each other, the cell kind of gets shorter. So like during cell crawling, the back of the cell will be pulled forward by these stress fibers. And what's creating that movement, whenever you have actin, the thing that creates movement is myosin. So here you can see two myosin molecules. Each myosin molecule is a dimer. And again, here's your coiled coil structure, very important. So you've got a myosin here that's grabbing onto this microfilament. You've got a myosin in the opposite direction here, grabbing onto this microfilament. So you can imagine if this guy walks along the microfilament in that direction, and this guy walks along in that direction, what happens, and because they're stuck to each other, they pull the microfilaments across each other. So very similar to muscle contraction. You've got myosin between two levels of microfilament, and they're pulling each other and contracting. Here again is that really cool immunocytochemistry. All the actins in red, and you can see stress fibers crossing the entire cell. Okay, so there's your parallel arrays, your anti-parallel arrays, and now the next thing are the gels. So we've got two different types of gels. One is this is the usual type of gel, which is found in the cortex. So basically, these are just long strands that are cross-linked, so that they form like a three-dimensional mesh. What's a good? Jello is a mesh. Jello is made from long sugar chains that you melt, right? And when you put gelatin in the hot water, it melts, and all these chains go straight. And then you let it cool, and then they cross each other and stick to each other, and you get this three-dimensional mesh. And the cell cortex is the same thing, except they're microfilaments. And every place they cross over, they're attached by a filament. So filament is a dimer protein. So it will bind to one microtubule, bind to another filament, and then that will bind to another microtubule. So that's holding them together. So you've got two filament dimers at this intersection, and that's keeping one microtubule attached to another. And that will surround the whole outside of the cell. That's the cortex. So we'll talk in a minute about the structure that's holding the plasma membrane together. And then this mesh is like a gel right underneath the plasma membrane, and it's giving it support. So that's holding the plasma membrane in place. Back up a tad. So there's your filament dimer again, attaching to two different microfilaments, and that holds them all together. And I'll show you another representation of a mesh in a minute when we talk about the cortex in more detail. So the fourth and final type are the branched microtubules. So you see these in lamellipodia. So the lamellipodium is like basically the plasma membrane extends out to grab forward when the cell is crawling, to pull itself forward. And the extension of that lamellipodium has to do with polymerization of these microfilaments. So they're getting longer and longer and pushing the plasma membrane forward. And part of that is branching. So back here in the cortex and in the other ones, there's no branching. They're all just straight polymers. And here they start to branch. I'm going to talk about that more later, I guess. This is just the general structure discussion. Okay, the next thing to talk about, so now we're going to talk about actual structure. So that if you look at the way the lecture's put together, we're going general, more and more specific. So we talked about just the structure of microfilament, 4 different things that microfilaments are used for, and now we're going to see them actually used in the cell and what they do. So one thing that microfilaments are important for are the structural elements that are going to hold the cytoskeleton to the plasma membrane. And this is what's actually holding that plasma membrane in place. You've got 2 different transmembrane proteins. And the idea with these transmembrane proteins is they've got at the outside of the cell and inside of the cell, all the amino acid side chains of the amino acids in that structure are hydrophilic, right? Water loving. So they stick out and into the cell. Now, all the amino acids in the transmembrane regions are going to be hydrophobic. So these are going to want to be in the membrane. These are going to want to be out of the membrane, which means this protein is really stuck in that membrane. You can't pull it out easily. So, because if you pulled it out, it would expose the hydrophobic amino acids to the aqueous solution outside, and it's more stable if it's in, so lower energy if it's in the lipid environment. So this protein is stuck here in the membrane, as is this, and this is what's going to anchor everything to the membrane. Now, what anchors these to the cytoskeleton are these other proteins here. So you've got ankyrin, which is a protein that binds to these transmembrane proteins and binds to spectrin. And spectrin is a protein then that's going to bind to small actin chains. And band 4.1 is going to help stabilize that spectrin-actin interaction. So in this figure, I don't like this figure. This is the one from the chapter that we're doing right now. They show band 4.1 binding to this glycophorin. I'm not sure it actually does that. There's another picture I'll show you in a second where it's not. What's the most important to remember with band 4.1 is it's attaching spectrin to the actin. And now that you've got these short actin bands, you can attach the gel cortex to those actin bands, and that holds the whole thing in place. So if we look at this, so this is the basic structure, right? Without even this, just transmembrane protein, ankyrin, spectrin, band 4.1 and actin. That's your basic structure. And you can imagine that just repeating over and over again across the underside of the plasma membrane. So kind of in 2 dimensions all around the cell, this structure is repeating over and over again. So here's your band 3. Band 4.1. Yeah, okay. So band 3, transmembrane protein, it binds to ankyrin, which binds to spectrin. Then you've got 4.1 binding spectrin to the microfilament, which binds to another spectrin, to another ankyrin, another band 4.1, or another band 3. So you can see the same structure repeating over and over again. Here is an electron micrograph of this. Okay, here's your actin and band 4.1, spectrin molecules going this way, and chirin and spectrin. Here's another drawing. So this drawing is from a really cool video I'm going to show you in a second, Inner Life of the Cell. I'll show you part of it anyway. So again, you can see the spectrin dimers, again, coil. Hello, Dr. Slish, you keep saying that, I'm sorry. So your spectrin coil, and then all the other proteins holding it together. Okay, cell cortex. So cell cortex then is really kind of like a gel, really, because it what I mean by gel is it's a structure that slows the movement of everything through it, right? Just if you've ever done an agarose gel for DNA, you pour that jelly out and you put your DNA in it, and small pieces of DNA can go faster through that mesh. Bigger ones get slowed down more. So that's kind of what's going on here in this mesh as well. It's hard to explain what I mean by a mesh, but I want to look. Let's watch this Inner Life of the Cell. I just want to watch a short part of it, and you'll see what I mean by these structures we just talked about. And I know I have to change my share. It'll just take a second here. Come on, baby. Share. This, I'm going to skip ahead. Some really cool stuff here. We'll watch this whole thing eventually. Oops. We'll watch this whole thing eventually. A lot of it we haven't talked about yet, so just give us a minute. I don't know if you're going to be able to hear this. Doesn't matter. The audio is just some music. So this is a receptor in the membrane. Here's your spectrum. Okay, and it's attaching different molecules together. And there's the structure I was showing you with that electron micrograph. Spectrin tetramer, and here you've got your actin and your band 4.1 holding them all together. Okay, now we're going into the cell. Who is that science teacher, Miss something with the magic school bus?
Oh, Miss Frizzle.
Miss Frizzle. I'm Miss Frizzle today. We're going to go into the cell. Okay, so there's your spectrin attaching to transmembrane proteins. Okay, here's your gel mesh. Watch this. Watch us go through this. You see, it's like three-dimensional cages. And that is the cortex. Let's go through that again, just for the hell of it. Where's Arnold? He's supposed to be in the bus. What's he doing now?
He finally decided to stay home today.
Good, he's a whiner. So there's your gel mesh. Okay, now we're inside the cell, and these are microfilaments here. You can see filament cross-linking microfilaments. Those yellow things are filament. Here's polymerization of a microfilament, which it usually isn't spontaneous. Usually there's ARPs controlling it, and I'll show you that later. But there's polymerization at that plus end. Here's a molecule called gelsolin. So gelsolin's going to grab this in the middle, break in the middle of the strand, and then the thing starts to fall apart. So gel solid is important for taking these microfilaments apart. And here's a microtubule polymerizing. Again, it's controlled by maps, but just so you can see it, they're showing it with only the microtubule, only the tubulin. And here's catastrophe. They're just peeling off. All right, and now this is chapter 14. We'll talk about the motor map dragging your vesicle along that microtubule. And in the foreground, we still got microfilaments and filament holding it together. Okay, so hopefully you have a sense now of what the cortex looks like. It's kind of like cages stacked on top of each other, and that's holding the membrane together. Okie doke. What else do we got? Okay, hang on. What's next? Right there, mister. Okay, branch networks. Oops, I'm going backwards. Okay, branch networks are found in the lamellipodium. So this is a crawling cell. We'll talk about this in the next chapter, but the cell here reaches out with this foot. That's the lamellipodium. And you can see it's a branch network of microtubules or microfilaments in that lamellipodium. Here's from a different angle. Now at every one of these sections here, in order to get a new microfilament to grow off of the middle of a chain of microfilament, you need to put a new nucleation point there. And that's what these proteins do, the WASP and ARP2 and 3 complex. So these are two different proteins that are always bound together, ARP2 and 3. So what happens is WASP, when it gets activated, will grab an ARP2/3, drag it over and drop it down in the middle of a microfilament, and that provides a nucleation point. The WASP will go off and find another ARP2/3, but once that nucleation point is there, boom, you can start polymerizing away from it. And profilin is part of what brings those monomers in to do that. Here's your capping protein, CAPZ, so that's what we'll talk about next. Okay, any questions or comments here? I feel like I'm going kind of fast, but I don't think this is all that complicated. Okay. Okay, polymerization. Okay, so how do we do this polymerization? Again, everything in the cell is controlled by proteins. So every step along this process will have proteins that are part of it. Skip over that one. Oh, here's your lamellipodium again. So you've got your usual cortex out here, and behind that cortex, you've got the branch network, and then you've got these stress fibers that are stretching across the cell. Blah, 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 blah. Come on, Dr. Slish, you said this already. Okay, so here is your cortex and blah, blah, blah. You can look at those later. Okay, proteins involved in polymerization. Number one, at the plus end, you've got CAPZ. So CAPZ is involved in microfilament capture, just like the tip proteins were in microtubules. So CAPZ binds to the plus end, and then it can't depolymerize. So during polymerization, it's got ATP, it hydrolyzes it to ADP, makes it unstable, but CAPZ holds onto it. And that CAPZ can also attach to other proteins of the cytoskeleton to hold those microfilaments in place. Gelsolin we talked about just a second ago. It can cut in the middle of a microfilament and then make it fall apart. So then now you've got unstable monomers and they just fall apart. Thymosin beta 4 is a protein that's going to stabilize these monomers when they've been freed. You don't want these monomers just floating around the cell going off in any direction, because then trying to polymerize would be extremely inefficient. Another protein then, profilin, this red ball here, is going to grab one of those monomers from from thymus and beta 4 and take it over to this bad boy here, formin. I think formin is like one of the coolest proteins. So formin binds to an open plus end and it's got these two arms hanging off here. And so formin will grab the cofilin ADP, cofilin complexed with actin with ATP on it, grabs that, brings it in, and adds it. So it's just kind of like two hands going up, putting new monomers on and stretching out that plus end. There's a really cool animation about this in our Moodle page. See if I can find that quickly. Formin profilin. Oops, let me pause this for a second. New share. It looks like it's grabbing food and eating it, shoving it down its throat. So the little yellow things are the ADF cofilins. Oops, that was fast. That was way too fast. I'll play that again. No, I'm sorry, they're the profilin. So the little yellow balls are profilin, bringing in monomers. I could watch this all day. I'm not going to, I'm getting paid for something else.
So it just floats around until it finds specific monomers and then it begins?
What, the formin?
Yeah.
I think this particular video is kind of like in vitro. So if you put this together in vitro, that's what it would look like. But normally it's going to be near and then attached to the plus end of microfilaments. So it's not just around, it's going to be controlled.
Okay.
And when you think about it, like the plus end of a microfilament will be capped with cap Z. So formin's just sitting around doing nothing. When cap Z comes off, formin grabs it and then it starts polymerizing it. So we'll see that cap Z thing in a second, I believe. All right, let's. Damn it. Damn it to hell. Oh, shoot, wrong class. There's another one here that I haven't watched in a year. Let's see what this one looks like. Okay, so there's something we haven't talked about yet. Let's continue on and then come back to this one, because I think we can probably tie it all together with this animation. Okay, so that's how you polymerize microtubules. Now, the next question is, how is that regulated? Because it's not just happening randomly, right? We're in a cell. We don't mess around when we're in a cell. Everything's controlled. Unlike this class, everything's in control. Okay, and it's controlled, oops, shit. It's controlled by activating G proteins. I don't know how many of you have heard of G proteins. I talk about them, used to talk about them in Bio 101 when I talk Bio 101, but they're basically intermediary proteins between a receptor and an effector, or the protein that's gonna make changes in the cell. So this is all about cell crawling. So why do cells crawl? One reason why they'll crawl is they'll sense some kind of chemoattractant. So imagine you've got some kind of a bacterial infection underneath your skin, right? So those bacteria are giving off chemicals, and you'll have white blood cells like macrophages and things going along inside the capillary, and they'll sense those chemicals, and then they'll go towards that chemical and grab those bacteria and eat them up. That's chemoattraction. Or you can imagine a gradient of a hormone. Maybe there's a tissue that's trying to attract these cells over here. And so it gives off a hormone, and these cells sense that hormone and they go in the direction of more and more concentrated hormone. That's typically what cell crawling is about, cells going towards some source of chemical. So how this works then, let me bring up my notes because I don't have a really good, couldn't find a good, a good figure for this. I had an animation that I did in Flash, but Flash is no longer supported by most browsers, so I couldn't do that. Couldn't play it. Oh, actually, I might be able to play that for you. Anyway, the signal is a chemoattractant. This chemical is usually a gradient that comes from some source and leads the cell towards its destination. So the chemical binds to a receptor at the cell surface, and that receptor then activates a G protein, and oftentimes a kinase will get activated, and membrane phospholipids can be involved. So these are just some of the components of this. So a G protein, what the hell is a G protein? Well, I'm glad you asked. Okay, so here is a G protein. I'm trying to put your faces out of the way. Where is it? Rho is a G protein. So there's 3 G proteins that will be involved in this process. RAC, Rho, and CDC42. Remember those names. I use them over and over again because they're really important for this, and we'll see them again later in the class, in the next chapter. And then when we cells start dividing, we'll see them activated again. These are small monomeric G proteins, RAC, Rho, and CDC42. And these proteins will be activated by a receptor. And this receptor is kind of generically called a GEF, guanine nucleotide exchange factor. So when the chemical binds to the receptor becomes a GEF. So what the guanine nucleotide exchange factor does then is it will interact with the G protein and cause the G protein to release GDP. Once it's released GDP, it will pick up GTP. Okay, so now it's become active. So the receptor is causing that G protein to release GDP, it picks up GTP. And then this will go off and activate some things and cause the actin microfilaments to change. Now, it's not going to stay on forever, right? With your car, whenever you've got a gas pedal, you got a brake pedal too. What's going to shut this off then is this protein called GTPase activating protein. So GAP is going to interact with Rho, and it's going to cause Rho to hydrolyze GTP. So GTP comes off, and then now Rho is inactive again. So now it's ready to be active again. So if the chemical is still out there, it'll get activated again by picking up GTP, and GAP will shut it off and turn it on and shut it off. So this is called G protein cycling. As long as you've got chemical out there that's activating the receptor, it'll keep cycling like this and causing actin remodeling. Here's another way of looking at it. Here's your GEF over here. It causes GDP to come off and picks up GTP. Your downstream effectors then are some of those ARPs that we talked about, of WASP and profilin and all of those things. And then you change actin cytoskeleton, and then it also gets turned off by GAP, GAP, GTPase activating protein, turns it back off. Okay, so okay, so here's an explanation of how that works with a couple of examples. Your small monomeric G proteins then are RAC, Rho, and CDC42. And these are going to be activated by a GEF. Sometimes GEF is called GNRP. I think this is an older name. I'm not sure people are using this anymore, but if you see that, it means the same thing, guanine nucleotide release protein. And again, this whole GEF thing is just a general term that we use for anything that will activate a G protein. And in this case, we're talking about an active receptor activating G protein. Typically, that's what it is that activates it. Okay. So this GEF then causes the G protein to release GDP and pick up GTP, and then the G protein is active. We then activate the active G protein then activates other signals and transduction proteins. So downstream effectors in that figure. These downstream effectors then will cause the remodeling of the cytoskeleton. And one example, we got two different examples here. One is GEF activates CDC42. CDC42 then activates WASP. WASP transfers ARP to ARP23 to the microfilament and nucleates a new branch. So WASP is sitting around there doing nothing until CDC42 gets activated. That activates WASP, and then you get the branching happening, and you can get a lamellipodium. Another example, more complicated, is your receptor activates something like CDC42 again. That activates a protein called PIP kinase. I should just say PI kinase, sorry. PI kinase. And PI is phosphatidylinositol. That's a membrane phospholipid. Something like 5% of all the phospholipids in cells are PI. You also have PS, PE, and PC. So let's see. So PI is just another membrane phospholipid sitting around. CDC42 activates PI kinase. Being a kinase, it puts phosphate on PI. So PI then becomes PIP2. PIP2 then will bind CABZ. So remember, CABZ is on the plus end of the microfilament and stopping it from polymerizing or depolymerizing. Here, by producing this new phospholipid with a whole lot of big negative charges on it, it pulls that CABZ off the plus ends, and now Formin can grab that plus end and start polymerizing. Okay. Does that make sense? Questions, comments? Okay. And then I guess the last thing really kills me to let you go after 45 minutes, but I'm running out of things to talk about here. Come on.
I have a question.
Okay.
In your slideshow, it looked like you said RAC was the lamellipodia and the CDC42 was the filopodia.
Don't ask me that. I don't know the answer.
Well, I just wanted.
I know.
Okay, I just wanted to confirm because it looks like you have the opposite in your notes.
I know. You're absolutely right. And every year, I hope nobody notices. Every year somebody notices, but you're absolutely right. What she's saying is up here, I say CDC42 is going to activate branching, which is in lamellipodia. But then later I say CDC42, I think it's filopodia. I forget. Let me finish this last slide and then we'll come back to that because it's an enigma. It's been bothering me for about 10 years now. I don't know what the answer is, but it was smart of you to pick up on it. Okay, so let me just share that last slide and then we can talk about that issue. Before we do that, let me show you this animation. So this is the flash animation I had. Oops, hang on, you can't see it. Wait, are you seeing this? Did you guys see this like gray screen?
Yeah.
Okay. So here's the animation of that process we just talked about. So some chemoattractant binds to these receptors, and the receptor, I'm leaving a step out here, would activate a G protein, and then the G protein activates this guy, PIK. PIK then phosphorylates these PIs into PIP2s. And that will pull the CABZ off the plus end of the microfilaments. And now you've got plus ends that are capable of being polymerized. Okay, so that was the last thing I talked about. Now let's go to the last slide. So this is the complication that Lee was talking about here. Okay, so here's a cool experiment. What they did in vitro, right? We're doing this in a test tube. They took some cells and they serum starved them. And that means that none of the receptors are going to be activated. They've taken away all the growth factors. So you're going to have a cell that's in a quiet state, not in a crawling state. And so you can see this bright line around the edge. That's your cell cortex. And there are some other microfilaments within the cell, but for the most part, the main microfilament area is the cortex. Okay, now you're going to take a Rho protein and add it to a cell. And it's a Rho protein that's constitutively active. It's always going to be active. So we add that to these cells and we get this, stress fibers. The whole cell turns into stress fibers. With the same cell, then we'll throw in an activated RAC and we'll get lamellipodia all over the whole cell. The whole cell becomes a lamellipodium. And if we add just CDC42, we get all filopodia. Okay, so this is how they learned that Rho is important for stress fibers, RAC is important for lamellipodia, and CDC42 is important for filopodia. Okay, so let's get back to Lee's question because it's a smart question. I hate smart questions. Let me share this. Okay, so what she's saying, and this drives me nuts to this day, is when you look up CDC42, it does in fact activate WASP, which is important for branching. That's lamellipodia. But when you put just CDC40, oh, this is wrong. When you put just CDC42 in the cell, you get filopodia. I don't know why that is. I mean, it's two different things. I don't know. Maybe, I don't know. But anyway, what I want you to know, don't worry about which CDC, which thing activates WASP, but know this, that RAC is lamellipodia, Rho is stress fibers, CDC42 is filopodia. Actually, let me just take this out of my notes. Let's just say G protein. Okay, so I'm going to take CDC42 out of these because I don't know, it's confusing. There. Okay, we'll make it a little more general. But this is something that, I mean, I end up asking this on tests a lot because it's kind of like a good fill in the blank type question. Which of these activates lamellipodia, which stress fibers, which filopodia? So it comes up a lot. Okay, what's the last thing we do, Bailey, every day before we quit? What's the last thing we do? We do drugs. I love joking about drugs because it's like I was brought up in the 70s, and in the 70s it was like, try drugs, see what happens, you know. But once the 80s came, it became just say no. So I'm culturally different than you people. When were you in your teens? Like the 2000s? Yeah, you've got no hope. Okay. 2000 teens, really.
Oh God, I'm a little older, so I was in my teens and like, shit, I don't even remember. Oh, yeah, early 2000. Thank you, baby.
You're welcome, darling.
Old. Huh.
Okay. All right. Well, anyway, 3 drugs, cytoclasin, and you notice they're kind of like, I put them in the same order as the microtubule drugs in terms of mechanism of action. So cytoclasin A binds to the plus end and blocks polymerization, just like colchicine does, okay, but with microfilaments. Latrunculin A sequesters monomers, just like vincristine and vinblastine. And phalloidin will stabilize microfilaments so you can't depolymerize them, just like Taxol did with microtubules. Okay, so here's another kind of memorization challenge, because you'll probably see this on the exam. Know what drugs affect microtubules and what their mechanism is, what drugs affect microfilaments, and what their mechanism is. Don't confuse the two, because the way I like to ask questions to see if you really know your stuff is I'll say, you know, you're a cell biologist and you're working on microtubules. What would happen if you added phalloidin? And the answer would be nothing. Phalloidin doesn't do anything to microtubules. It affects microfilaments. Or I might say, you know, you're doing an in vitro microtubule assay and you add Taxol. What do you see? You see the microtubules getting longer and staying long, you know? What would happen if you added colchicine? They would fall apart. So know their mechanism. And if I give you a scenario where you've added this drug to something, you know, what would happen if that happened? Okay. Stopping the share. Sharon is Karen, right? Karen is Sharon. Okay. That's about all I have for today. You know, I hate to let you go early, but like I say, if we were in class, I'd cover the same amount of material, but it would be slower because I'd be writing it on the board. Any questions or comments? See, this is the honeymoon period. Until we have that first exam, everybody's smart, everybody's studying. Then you have the first exam, it's like, oh boy, we got work to do here. So I'm working on a game for you guys for this test. It's almost done. Tyler and I are working on these games. Hopefully I'll get that up for you next week and you can start playing that. But next week we're going to talk about the next chapter, chapter 14, and how the cytoskeleton is used for movement in the cell. I think we've got 3 chapters on that, or 3 or 4 lectures on that. Okay, questions, comments? Okay, I'm gonna let you go. Goodbye.
Bye.